Nervous stimulation excites a skeletal muscle cell. An action potential will trigger the release of calcium ions into the sarcoplasm. Calcium ions bind to the regulatory proteins and trigger contraction. Structures within the skeletal muscle cell provide the mechanism for forcible contractions. Myofibrils, rod-like structures that extend the length of the cell, contain repeating units called sarcomeres. Each sarcomere contains many actin and myosin filaments. A single contraction is a cycle, the result of the filaments sliding over each other. Let's take a look at the actin filaments. The actin filament consists of actin proteins and regulatory proteins. The actin protein has a binding site for myosin molecules. Tropomyosin protein in the filament covers the myosin binding sites on the actin. A second regulatory protein, troponin, has a calcium binding site and is attached to tropomyosin. In the presence of adequate extracellular calcium, the troponin-tropomyosin protein complex uncovers the binding sites. Myosin can now attach to actin. This triggers a contraction. Myosin filaments consist of myosin proteins that have a head called crossbridge and a tail. The crossbridge has an ATP binding site. After ATP binds to a crossbridge, it undergoes hydrolysis to produce ADP and a molecule. This provides the energy to move the myosin protein. Myosin crossbridges attach to actin molecules or actin binding sites when bound together. The movement of the myosin protein or power stroke will move the actin filaments. The protein titin is a very elastic protein, allowing the skeletal muscle to return to its normal length after contracting or stretching. A muscle action potential triggers the release of calcium ions into the sarcoplasm, initiating a contraction cycle. A single contraction cycle consists of 4 steps. First, ATP attached to the myosin crossbridge must be hydrolyzed to provide energy needed to move the myosin protein. Second, actin and myosin filaments temporarily bind together. Third, the release of the phosphate molecule triggers the power stroke. The myosin filaments rotates, releasing the ATP. The force of the rotation slides the actin filaments toward the center of the sarcomere. Fourth, the myosin head and actin are detached when another ATP molecule binds to the crossbridge. This ends a contraction cycle. The cycle continues as long as ADP hydrolysis continues and nervous impulses continue to reach the cell. Let's see how this works. Contraction provides body movement or a force in transmitting through the series of structures to produce the movement itself. Movement by myosin filaments pulls actin filaments, sliding them toward the center of the sarcomere. The actin filaments are connected to the sarcolemma through dystrophin proteins. The shortening sarcomere causes the sarcolemma to move. As the sarcolemma is shortened, the entire muscle cell contracts. The more muscle cells that contract, the more forceful the contraction. The force of the contracting muscle is transmitted through the connective tissues of the skeletal muscle to the perosteum of a bone. The contraction proceeds in a manner that brings the insertion toward the origin, that is, insertion of a muscle toward the origin of muscle. In summary, the movement of actin filament sliding along the myosin filament is transmitted through a series of structures to move bones and body parts. This movement results in the most movable attachment side of the muscle, the insertion, moving toward the least movable attachment side of a muscle, the origin.